Our next goal is to learn how to put amino acids together, combine amino acids to ultimately make proteins. Before that, though, we're going to talk about combining amino acids to make a peptide. A peptide is a molecule that contains two or more amino acids. This is also true for a protein. So really the only difference between a peptide and a protein is just the number of amino acids that are incorporated in the molecule. A peptide is just a smaller version of a protein. So a peptide is two or more amino acids. They are connected or linked to each other with a bond that we call an amide bond. The amide bond is also called, so AKA peptide bond. And what we're going to do is let's just draw a really small and simple peptide. So for our amino acids, we're going to have our first amino acid. We're gonna draw these when we're forming peptides. Our amino acids are always going to be drawn in the zwitterion form. So we're not thinking about pH at this point. We're just drawing everything as a zwitterion. And in this section, you're going to see why I prefer to draw my amino acids a little bit differently than the way that they're drawn in the tables, because drawing them in this format with the hydrogen up top and the acid group on the right and the amino group on the left is going to make this really easy. So here's our first amino acid that we've drawn. This is called alanine. Its abbreviation, 3-letter abbreviation is ALA. Its 1-letter abbreviation is just a capital A. That's going to become relevant later. And we are going to combine it with amino acid number 2 in the zwitterion form, so COO minus on the right-hand side and NH3 plus on the left-hand side, and its R group is going to be CH2SH. So this amino acid is cysteine. A 3-letter abbreviation is CYS. And its 1-letter abbreviation is just a capital C. So when these two amino acids come together to make a peptide, they connect at the amino group of the right hand with the acid group of the left-hand amino acid. So these two that I've highlighted in blue, these are the functional groups that are going to connect the two amino acids. So when we start drawing the peptide, we can take, for our left-hand amino acid, for alanine, everything that I'm shading in red or green is going to be intact in the peptide, like no changes to it at all. So we can start by just drawing that stuff that I shaded in green, because we know that it's not going to be changing. So we have NH3 plus, we have our carbon in the middle, and then we have our R group CH3. So, and I'm going to highlight that green so we can kind of keep track of it. So there's the portion of alanine that is just, nothing is happening to it at all. So for the linkage, the amide bond or the peptide bond between these two amino acids, the alanine is going to contribute its carbon and one of the oxygens in the COO group. So this right here is going to be, I'm going to actually change my color coding over here. What I've shaded in blue is this much of the alanine molecule right here. And that is it for alanine. That's all that it's going to contribute. So the next thing, that bond that I just drew right there, this is going to be the connection between the two amino acids. So this is going to be our amide bond or our peptide bond that connects the two. And we're ditching that last oxygen on alanine. It is going to show up later, but we're ditching it for now. The next thing that we're going to connect to is the nitrogen of the cysteine. So I'm going to highlight that nitrogen right there that guy highlighted. And, ooh, I need only one of its hydrogens. So how could I draw that? Let me change the way I'm drawing the 3 hydrogens on this guy. I'm going to actually draw them out. So I need 1 nitrogen and 1 hydrogen only of the cysteine, and that's right here. We'll get that in yellow. And then the rest of the cysteine molecule, what color do I have left? I have red. The rest of the molecule is going to just be left alone. So it's going to come across into the peptide without any changes to it at all. So there is the rest of that amino acid, totally unchanged, highlighted in red, and there we have it. So that's all there is to it. Now the last thing we need to do, I'm going to need a different color here. The 2 portions of the amino acids that were not incorporated in this peptide, so we have the oxygen from the alanine, and we have the 2 hydrogens from the cysteine. Maybe if you're very clever, you might be realizing an oxygen and 2 hydrogens, what does that make? That makes water. So every time we connect 2 amino acids together to make a peptide, for every amide bond or every peptide bond that we make, we are also producing 1 water molecule. So we're going to get 1 H2O for every amide or peptide bond. And again, that water, that H2O, is coming from the oxygen and the hydrogens from the original amino acids that are not being incorporated in the peptide. So this whole thing together right here that we've made, this guy, is our peptide. It is made up of, this particular one is made up of 2 amino acids. Its name is ALACYS, which comes from the names of the 2 amino acids that are incorporating that peptide with a dash in between the abbreviations. Or we could also call it AC, which comes from the 1-letter abbreviations for the individual amino acids. If we're using the 3-letter abbreviations to name the peptide, we will put a dash in between each 3-letter abbreviation. So there's a dash when we are using the 3-letter abbreviations. But if we are using just the single-letter abbreviation, no dash. It is important that you write the peptide's name in the correct order. So for this one, its name is AC, not CA, or ALACYS, not the other way around. We always write the amino acids in a peptide from the left side to the right side as it is drawn. So we always want to write it from the left side to the right side. Now you might be saying to yourself, left versus right, how am I going to know? I could have easily drawn this the other way around and got my lefts mixed up and my rights mixed up. So a better way to say it than from left to right is that we always name an amino acid from the what we call the N terminal to the C terminal. So we are going to write the name from the N terminal. So we start at the N terminal and then we go to the C terminal. Now, if you're drawing your amino acids in this form, then the N terminal is always going to be on the left and the C terminal is always going to be on the right, and there isn't going to be any tricks involved. But if for some reason things do get kind of tricky, what's the N terminal and what's the C terminal? Well, that is the N terminal. So if we're looking at the amino acid, it's going to have two endings to it. One of the ending on one side is going to be the NH3+, and we're going to call that the N terminal. And then what about the C terminal? Right there. The other possible ending for the amino acid is the COO-. C for carbon, this is the C terminal. So when we're writing the name of an amino acid, we're always going to find the ending that contains the nitrogen, the N terminal. We're going to start there, and we're going to name our amino acids in order proceeding from N to C. So now what we're going to do next is practice drawing a few different amino acids. And I think you're going to find that this is pretty straightforward once you get into the pattern of it. So we're going to do one here in our notes, and then we're going to go to a worksheet and practice some more. We are going to start by drawing the structure of a peptide called VALSERASP. Or with the one letter abbreviations, its name would be VSD. Now, in order for you to be able to do this, you need to have handy a table of amino acids. So you need to find the table of amino acids that you're using. Just find one online, something that looks appealing to you. I did link one of them on Canvas, but you can use whichever one looks good to you. And you need to have that close by so that you know what the R groups look like. Before we get into our R groups, though, we're just going to create the framework for this peptide. So we know that the peptide is going to be drawn from the N terminal. That's the very first thing that we're going to have on the very left-hand side. And for our first amino acid, we are going to have that carbon that has the hydrogen sticking straight up, and that amino acid's R group, which we'll come back in and fill in later. So there's going to be something down here that makes it the VAL amino acid. And then for this amino acid, it gets to keep on its right-hand side its carbon and one of its oxygens. Let's look back up at our reference here. So what we have shown up here in blue, it gets to keep the carbon and one of its oxygens, and that's it. And the next thing that happens then is that we now connect to the next amino acid in the chain. So this is where we have the nitrogen and one of the hydrogens from amino acid number 2. So amino acid number 2, the nitrogen with only one of its hydrogens. The carbon in the center with the hydrogen on it, and this is where we will have that amino acid's R group, so SER, whatever that is, will get drawn in right there. And that amino acid gets to keep its, of its acid, it gets to keep carbon-oxygen double bond, but that's it. And now we connect to the last amino acid in the chain, ASP. And because this is the last amino acid, it gets to keep its entire acid group, so COO-. So what we have here, let's put some other markings on this. This right here is going to be our VAL amino acid. And this chunk right here is SER. And this chunk right here is the ASP. And this is our C terminal. And this is our N terminal. When I'm drawing a peptide, I like to start by just drawing this. I want to draw the framework for as many amino acids as I'm being asked to include, 3 in this case, get the pattern down, and then I go to my table and find the structures of the R groups for each one of these amino acids. So now it's time for you to go to your table, and the first thing you have to do is find valine, VAL, and you'll see that that guy is a CH with a couple CH3s hanging off of it. And now go find SER. Like maybe you need to be looking also at a table of 3-letter abbreviations. So you'll see that serine, SER, and its R group is CH2OH. And then the last one you want to look at, ASP. Find that guy. That is, ASP is aspartate, which is also known as aspartic acid. CH2. And because this is one of the acidic amino acids, you might be wondering, whoa, what am I supposed to do with the R group down here? How am I supposed to draw the R group on an acidic or basic amino acid? Let me tell you, it doesn't matter. Draw it how you see it on the table. That's handy. These types of problems are not asking you to be thinking about pH. So they're not, when these types of questions are being graded, they're really being looked at, you're really being evaluated at what you're doing right here in the molecule. Like, are you putting stuff together correctly? And not so much what's going on in terms of the charges of the R group. So don't stress about that at all. I advise you just copy it off of whatever table you have. That's going to be good. Let's go to our worksheet and practice this some more.